Hi, and welcome to this online course on structural equations modeling and causal inference. The course is based on my book, Cause and Correlation in Biology, a user's guide to path analysis, structural equations, and causal inference with R. This is published by Cambridge University Press. This completely online course consists of over 18 hours of lectures in the form of videos. And these videos include practical sessions in which I do analyses in the R statistical environment, online courses for individuals consists of access to these videos for a period of 2 months, and you can follow this course whenever it's convenient for you. I also give online courses for groups that are 5 days in length, plus live sessions via Teams or Zoom. And I can also teach this course in person as a 1-week intensive course, and I've done this many times. If you go to the section on course details, then you will find information on the lectures. The first 2 lectures are freely available on YouTube. They're publicly available as well, so you can simply click on the links and you can view these 2 lectures free of charge. The following 8 lectures given once you've paid for the course, and I'll provide these links upon purchase. And if you go through this section, you'll find more information on the topics covered in this course, which is essentially the topics covered in the book, although there are 2 sections, a section on piecewise SEM and on equivalent models and AIC statistics, which are not found in the book. So, with that, let's start. Hi everyone, and welcome to my course on path analysis and structural equation modeling. My name is Bill Shipley. We'll be together for the rest of the course. Right now I'm sitting just outside my house in the woods. My house is in the eastern townships of southern Quebec, and I'm located about 200 kilometers south of the edge of the boreal forest. So the forests in my area are a mixture of hardwoods and conifers. If we were to go another 200 kilometers further south across the American border into the New England states, then the forest would be dominated by hardwoods, sugar maples and beech and so on. Now, where I am outside my house, the trees are predominantly conifers, balsam firs and white spruce. And the reason is because the soil here is rather shallow and rocky, and the water table is close to the surface. So the obvious cause of this change in vegetation going north to south is the climate. And additionally, there's more local causes related to the soil and the water table. There's a red squirrel who's sitting on the branch just above me, and he doesn't look very happy. I've invaded his territory. The reason why there are red squirrels on my property rather than gray squirrels is because the red squirrels are better adapted to eating the seeds from the cones of the balsam fir and the white spruce. In other forests in my area that are more dominated by sugar maples and beech, you find gray squirrels. Now, if you notice, the story I've just told you used a lot of concepts of cause and effect. So the vegetation is caused by climate and soils, and the presence of the red squirrels are caused by the presence of the conifers. And if you think about it, that's somewhat of a curious thing. Why is it that the language of science is permeated, dominated by the notion of cause and effect? But one of the main tools that scientists use to analyze their data, the statistical methods, does not have any concept of cause and effect. So the official language of statistics are probability distributions. And probability distributions refer to notions like dependence and independence, conditional dependence and conditional independence, marginal distributions and conditional distributions, and so on. And it's really not simple or easy to translate between the language of cause and effect, the language that scientists use, and the language of probability distributions that are used in statistical analysis. The purpose of path analysis and structural equations modeling is to provide this translation device between the language of causality that's dominating all of science, with perhaps the exception of quantum mechanics, and the language of probability distributions that are used in statistics and data analysis. So that's the topic of the course that you're going to follow. There are going to be sections of the course that are videos like this, where I'm giving you lectures explaining the concepts, and there are other parts of the course which will be online, live, in which we'll be discussing topics, answering questions, and I'll be showing you how to do various analyses in the R language. Now, it's a bit chilly out today, so I'm going to go back into my house, and I'll meet you there. So I'm back in my house and in the warmth. So let me tell you a little bit about the structure of the course, and then we'll begin our introductory lecture. So the course is completely online. There are going to be a series of recorded videos, like the one you're looking at now. These recorded videos are primarily designed to give you the theoretical information that you need. There'll be a little bit of R code that I'll be showing you on the videos as well. But most of the R code and most of the analysis will be explained live using Teams or Zoom or whatever program is most useful to you. These are the two main resources that you're going to need for this course, besides, of course, your computer and your Teams or your Zoom or whatever. The first, of course, is my book. You need this book because the course is primarily designed around it. Most of the information I'm going to be giving you in this course is found in the book, although some information I'll be giving you is information, ideas, methods that have been developed since the book was published. This book is available from Cambridge University Press, but you can also purchase it from Amazon or any other online bookseller. The second important resource is, of course, the R statistical language. You absolutely need this on your computer. I'm also going to be using the R Studio program, which interfaces with R and makes it easier to do the manipulations. If you want to use a different editor, then that's fine. Most of the functions are available on the CRAN website, so the main server for R. But a few of the functions I'm going to be using are ones that I've written and that aren't on CRAN, but you can download them from my GitHub site, whose address you see on the screen. So this course is about path analysis and structural equations modeling, whose primary goal is to test cause and effect relationships among several variables using observational data. Now, you can certainly analyze experimental data using these methods as well, but the primary purpose is to be able to test cause and effect hypotheses when you don't have access to experimental manipulations. Before we talk about path analysis and structural equations modeling, though, we first have to take a step back and look at how scientists have historically tested causal hypotheses using experiments. Now, historically, there have been two different types of basic experiments that allow us to test cause and effect hypotheses. The first, historically, is called the controlled experiment. The second, which is inferentially much stronger, the gold standard, is the randomized experiment. I want to talk first about the randomized experiment, and then I'll go on to talk about the controlled experiment. So the randomized experiment was formally developed by the person you see on the screen, R.A. Fisher. R.A. Fisher was one of the great statisticians and theoretical evolutionary biologists of the 20th century. And the logic and methodology of the randomized experiment was presented in his 1935 book, The Design of Experiments. Now, he already was using and was thinking about randomized experiments before that, so in his 1925 book, which deals more with statistical methods rather than experimental design, the randomized experiment was already being used. So the randomized experiment was developed sometime in the beginning of the 20th century. And if you notice, this is rather a curious thing because scientists have been testing, or at least they thought they were testing, cause and effect hypotheses long before the beginning of the 20th century. And they weren't using randomized experiments. But as I said, the randomized experiment is in some ways the gold standard now for testing cause and effect hypotheses. So let's look a little bit about how one goes about conducting a randomized experiment and the logic behind it. So since Fisher initially worked at the Rotherstam Agricultural Research Station, let's imagine an experiment which might have been done to test whether a new nitrate fertilizer increases the yield of wheat. So we're going to go through the steps, and then we'll look at the logic and why combining the logic with the statistical analysis we can draw conclusions about cause and effect. So the first step is in the springtime before the seeds have been sown or any manipulations have been done. You divide the field into a series of plots, or more formally experimental units. So the experimental unit is the physical thing whose attributes you're going to be manipulating or adding or subtracting treatments to. And so we separate the field into these plots or experimental units. And then the second step is to randomly allocate the treatment or the control to each plot. So one way to do this is to simply take a little piece of paper, one for each plot, and then write a T or a C on it, and then randomly draw these pieces of paper from a hat after thoroughly mixing it. So notice the idea here is that you're randomly allocating the treatment or the control to each plot. And because this is a random allocation, we can assume that whatever is happening between the variables in our experimental units, that the addition or not of the treatment has not been determined based on these attributes. So we randomly allocate 15 of our 30 plots to receive the treatment. The treatment is the nitrate fertilizer dissolved in water, and we apply a constant amount of water to each of our treatment plots. And then the remaining plots, the controls, receive the same amount of water, but in which the fertilizer hasn't been added. So the next step then is to allow the wheat to grow during the season, and then in the fall we harvest the wheat. And imagine that this is the result that we get. So in the control plots, we have weighed 55 grams of seed that's been produced with a standard error of 6. And in the treatment plots, we notice that there are on average 80 grams of wheat seeds produced, again with a standard error of 6. So the next step is to do a statistical analysis. And this statistical analysis, as you'll see in later slides, isn't telling us anything about cause and effect. It's simply telling us about the chances of observing statistical differences between numbers. So a statistician would tell us the first step then is to establish your null hypothesis. And the null hypothesis in this case is that the treatment and the control plots all come from the same statistical population. In other words, there's nothing different between the treatments and the controls except for random sampling fluctuation. So the theoretical mean is therefore the same in the two groups, even though the sample means can differ because of sampling fluctuations. So now we want to know if this null hypothesis is true, and in the statistical population, the means of the treatments and the controls are the same. What's the chance of observing purely by chance, by random sampling variation, a difference equivalent to the difference between 55 and 80? And so we would do a t-test, calculate the t-value, which in this case is 7.91, and if the null hypothesis is true, then this large t-value would have arisen by chance. And so the next step is to calculate the probability of observing at least a value of 7.91 purely by chance, assuming that in fact the population means are the same. And when we do this, we find a very small probability, 5 times 10 to the minus 8. And so we can conclude as a statistical conclusion that the plots receiving the nitrate fertilizer really did produce more wheat seeds than the plots not receiving the nitrate fertilizer. The reason being that if we don't conclude this, then we have to accept that we are in the presence of an extremely rare event, something that would happen less than 1 in a million times. And so we prefer, therefore, to conclude that the null hypothesis wasn't true, and in fact, the control and the treatment plots come from two different statistical populations. Now, at this point, I want to pause for just a second and point out an important distinction between statistical analysis and causal analysis. Now, in the story I've just been telling you, our causal conclusion, that is, that the added nitrate fertilizer increases the yield of wheat, doesn't come from the statistics. The causal conclusion doesn't come from the fact that our t-value is significantly lower than our significance level. After all, you can do a t-test on any two groups. You can ask whether the means of any two groups are different with a t-test, and those two groups don't have to represent treatment and controls in a randomized experiment. The t-test is simply telling us that the association between increased yield and the presence of the nitrate fertilizer is not due to some random fluctuations in our data. So if the causal conclusion doesn't come from the statistical analysis, where does it come from? Well, let's go through the logic of the randomized experiment in determining whether a treatment has a causal effect on our experimental units. So the first point that we've concluded is that it's too unlikely that the treatment mean was larger than the control mean simply due to sampling variation. Therefore, the statistical test allows us to conclude that the mean really is larger in the treatment. The second step of the logical argument is to realize that there are only three, but not mutually exclusive, causal explanations for this statistical pattern. First of all, the added fertilizer could cause an increase in the yield. Logically, the increased yield could cause an addition of the fertilizer. And third, there may be some unknown common causes that are causing both an increase in yield and the presence of the nitrate fertilizer. So those are the three logically exclusive possibilities that we can imagine. Now, of course, we can exclude number 2 right away, because one of the properties of causes is that causes can't go backwards in time. So we can exclude number 2, but that still leaves possibilities 1 and 3. However, the third point is that we already know that the only cause of a plot receiving the added fertilizer, and that was the random number that we pulled from the hat. The only cause that determined whether that plot, that experimental unit, had the added fertilizer or not was simply whether the piece of paper that we pulled from the hat said T or C. This random number could not have been caused the increased yield alone. It's one of the properties of a random number is that it doesn't have any systematic cause. Therefore, when we put these points together, we can conclude that the causal structure has to be the random number caused the added fertilizer, and the added fertilizer had to cause the increase of yield. We can exclude number 3 because the only cause of fertilizer was the random number. And if that random number was the unknown cause, then that random number couldn't have also increased the increase in yield directly. So that's the logic of the randomized experiment. So we've looked at the logic of the randomized experiment, but it's important to point out some of the limitations of the randomized experiment, because these limitations are important and they're quite common. So one limitation is simply ethical limitations. There are many situations in which it's unethical to randomly assign the treatment variable to experimental units. You'll notice on your screen the picture of Ronald Fisher smoking a pipe. Ronald Fisher was a smoker. And in fact, Fisher wrote a letter to the British Medical Association objecting to their conclusion that smoking causes cancer. And his objection was that this conclusion wasn't based on randomized experiments. And yet, if you think about it, it would be pretty much impossible to run a randomized experiment to test whether smoking causes cancer. You would have to get a group of young people before they begin smoking, randomly assign these young people to a treatment or a control group. If the person happens to land in a treatment group, then this person has to agree to start smoking and to smoke for their entire life until we find out whether the person develops lung cancer or not. And the control group, the people would have to agree to never smoke. If you think about it, it would be pretty much impossible to run an experiment like that. There are important ethical limitations in ecology as well. So if we're studying the causal effects of some environmental factor on a rare species, it would be unethical to randomly assign individuals of this rare species to treatment and control groups to see whether those individuals would die or not. No research granting agency would ever agree to such a thing. So these are important ethical limitations. There are also some important scale limitations. Now, the logic of the randomized experiment that I just explained applies only to the scale at which we've done the experiment, that is, to the scale of the experimental units. So in the example of the fertilizer in the field, the experimental unit was 1 plot. Imagine that you wanted to test the hypothesis that an increase in atmospheric carbon dioxide increases net primary productivity of a forest. In this case, the experimental unit would have to be the entire forest. It wouldn't be acceptable to run the experiment on a single tree or a few trees and then extrapolate up to an entire forest, because the experimental unit of a forest includes a lot more things than you find simply in a single tree or in a few meters of forest ground. So these scale limitations are really important. And imagine if we wanted to test whether increased carbon dioxide concentrations in the atmosphere are causing increases in global warming. In this case, the experimental unit is the entire Earth, and we only have one Earth, so we couldn't possibly run a randomized experiment like that. So these sorts of scale limitations are really common, and they have really important limitations in terms of making cause and effect conclusions from statistical experiments of the randomized type. Now, there's another limitation of the randomized experiment. This limitation is a little bit more subtle, but it's really, really common and important. in ecology and evolution. And that relates to the way that we manipulate the treatment variable. So the logic of the randomized experiment requires that we be able to randomly assign different levels of our treatment variable independently of any attributes of the experimental unit. So in our fertilizer experiment, the treatment variable was the amount of nitrogen that we dissolved in the water and added to our plots. And we can certainly randomly allocate the amount of nitrogen that we add to our plot. But imagine we wanted to test a more complicated causal explanation, the one that you see on the screen. So we want to know whether the increased nitrogen fertilizer that we add causes an increase in nitrogen absorption by the roots, which then causes an increase in photosynthetic enzymes in the leaf, which then increases the amount of photosynthesis and carbon that's fixed by the leaves, which then increases the seed yield of the plant. Now, if we wanted to test any of the causal links inside the box, we have to be able to randomly assign different levels of that variable independently of any other attribute of the experimental unit. So, for instance, if we want to know whether increased photosynthetic enzymes increase carbon fixation, we have to be able to randomly assign different amounts of photosynthetic enzymes to the leaf directly, independently of any other attribute of the unit, of the experimental unit, and then see whether the carbon fixation increases or not. But we can't do that sort of an experiment. You can't randomly add or subtract photosynthetic enzymes to a leaf without simultaneously changing everything else. Now notice that when we added the fertilizer, we could certainly see whether the fertilizer increases photosynthetic enzymes. We could certainly see whether the nitrogen fertilizer increases the amount of carbon fixation. But we can't see whether nitrogen fertilizer that was added caused increased carbon fixation by increasing photosynthetic enzymes. Perhaps nitrogen increases photosynthetic enzymes, perhaps it increases carbon fixation, but that photosynthetic enzymes have no relationship to carbon fixation. So we can't do that type of an experiment. And this limitation is extremely common in ecology and in evolution, simply because the experimental units that we will look at, plants, animals, ecosystems, are integrated wholes, and we can't randomly manipulate parts of that whole without changing everything else at the same time. So the randomized experiment is very powerful, but it has some important limitations. I've called it the gold standard, and it is, because when the randomized experiment can be done properly and correctly, it provides the strongest inferences about cause and effect. But it's very often limited. It's limited by ethical constraints. It's limited when the scale of the phenomenon is too large or too small to be manipulated because of time or money or other reasons. And it's limited when the question involves causal relationships between attributes of the experimental unit. So if you think about it just a little bit and you apply it to the sort of questions that you're studying, I'm pretty sure that you will be able to quickly identify examples from your studies where you can't really do a randomized experiment. So I said before that I'm going to talk about randomized experiments and controlled experiments. I talked about the randomized experiment first because it's the gold standard, but remember that the randomized experiment only began to be used by scientists at the beginning of the 20th century. But certainly science and the study of cause and effect go back well before the beginning of the 20th century. And before the 20th century, randomized experiments were not used. Instead, what was used was the controlled experiment. No, I'm no historian of science, and so I can't pinpoint exactly when the controlled experiment was first introduced. I suspect it was never formally introduced. It was a method that was developed and discussed and modified by scientists as they proceeded. But I'm going to illustrate it by the example of the man you see on the screen, Claude Bernard, who was an experimental physiologist. He was one of the first experimental physiologists, and the first one to really formally and explicitly describe his logic in using a controlled experiment to derive cause and effect conclusions. English translation of the book is called An Introduction to the Study of Experimental Medicine. And I'm just going to give you a quick quote here. So in the English translation, the quote is to say that, indeed, proof of a given condition always precedes or accompanies a phenomenon. So here he's talking about a correlation, of course. And he says that this given condition does not warrant concluding with certainty that a given condition is the immediate cause of that phenomenon. So here he's simply saying that observing an association, a correlation, is not evidence in itself of a cause-effect relationship. He goes on to say, it must still be established that when this condition is removed, the phenomenon will no longer appear. In other words, he's saying you have to then experimentally control the variable of interest, the condition, and see whether, once you've controlled that variable and prevent it from responding to its natural causes, whether the patterns of association change or not. So I'll describe one experiment he did. These are rather gruesome experiments, in fact, in which he used live dogs, which he opened up and then did these manipulations on them. So these are not the sort of experiments that anyone could do today. In the experiment I'm going to discuss, he was looking at the functioning of the kidney. He was trying to understand what the kidney does and how it works. And he noticed that the color of the blood coming into the kidney through the renal artery is different than the color of the blood that leaves the kidney through the renal vein. And he could also observe whether the kidney was actually being active or not by seeing whether urine waste was being produced and excreted through the ureter. And so he hypothesized that the color, change in the color between the blood entering and leaving the kidney, was due to the kidney removing wastes from the blood. So he hypothesized then that the blood entering the kidney through the renal artery has waste in it. When the kidney is active, that is, when it's producing urine, it then removes these wastes, which then changes the color of the blood when it exits through the renal vein. And to test this hypothesis then, he would experimentally control or fix the variable whether the ureter was producing waste or not. He did this, I believe, by tying off the ureter to prevent urine from being excreted from the kidney. And so what he would do then is to physically, experimentally control the active or inactive state of the kidney by tying off the ureter or not. And he observed that when he prevented the ureter from excreting urine, the color of the blood entering and leaving the kidney didn't change. And when he allowed the ureter to function normally, the color did change. So in this case, it's called a controlled experiment because he was physically controlling a variable of interest, that is the active or inactive state of the kidney, and then seeing whether the patterns of association between the other variables change as predicted by his hypothesis once he actually conducted the experiment. So this is the logic of the controlled experiment. 6 steps. The first step is to hypothesize how you think the causal links work between your variables. So you saw the example in the slide before about the color of the blood, the active or inactive state of the kidney, and the color of the blood leaving the kidney. Step 2 is you predict how the variables will be associated or correlated if the hypothesis is true. And then you predict how those patterns of association will change as you control different variables, and then assuming your hypothesis is true. Another word for control is to fix or to hold constant. These are pretty much synonyms, which I'm going to use interchangeably. So when I talk about controlling or fixing or holding constant a variable, this is what I mean. So after you've devised these predictions based on your hypothesis, you then conduct the experiment by physically controlling these different variables and then comparing the observations of these associations to the predictions that your hypothesis produced. So you want to say, does the color of the blood change if I control and prevent the kidney from producing urine? And do the color return if I then allow the kidney to function naturally? And the final step then is that you reject your hypothesis if the observations don't match the predictions. And if the observations do match your predictions, then you can provisionally accept your hypothesis. And the key word here is provisionally, because the controlled experiment is not as strong as the randomized experiment, mostly because you can never exclude the possibility that you may have missed certain variables. There may be certain variables that you didn't control or are even aware of, which may generate equivalent predictions to the one that you hypothesized. So it's not as strong as the randomized experiment, but it is the method which most scientists used before the randomized experiment was produced. And if you think about it, this is still the experimental method that is very commonly used when you can't do a randomized experiment. So we've talked about randomized experiment and the controlled experiment. And the key notion in a controlled experiment is the idea of being able to reach into your system, physically take control of a variable to prevent it from changing, and then see how the patterns of association, the patterns of correlation between the remaining variables change once you've taken this physical control. Now, you might be wondering what all of this has to do with path analysis and structural equations modeling. After all, path analysis and structural equations modeling are designed for making inferences about cause and effect when we can't or we haven't conducted controlled experiments or randomized experiments. Well, the key point of a controlled experiment is the notion of control, of being able to prevent a particular variable from changing as it normally would. It's not physical control. In other words, control is the key word, not physical. The idea of path analysis structural equations modeling is that we replace physical control of variables with statistical or observational controls. Now, physical controls and statistical controls are not at all the same thing. You mustn't get them confused. And it's going to take us an entire second lecture to explain really the differences and the similarities and how you can link these two together. So you mustn't for a moment think that physical control and observational control are the same thing. But there are similarities. And if we can figure out the relationship between physical control in a causal system and statistical control in a causal system, then we can use the logic of a controlled experiment by replacing physical controls by observational controls. And that is the key idea of path analysis and structural equations modeling. But just to give you a foretaste of what I'm talking about, imagine that you're studying bighorn sheep in the Rocky Mountains, and you've noticed a correlation, a positive correlation, between the body size of an individual in the fall and the probability of this individual surviving until the spring breeding season. So in the graph here we see that individuals who have 80 or 90 kilograms of weight have a higher probability of surviving to the spring than individuals who have 50 or 60 kilograms of body weight. The problem is that Rocky Mountain sheep are dimorphic species, so the males have on average larger bodies than the females. And so you might want to propose two different causal explanations for this pattern. One explanation might be simply that the two variables, body size and survival, are not causally linked. One is not causing the other.
It's that both of those are common effects of a single cause, which is the sex of the organism. So if you're a male, you have larger body size in the autumn, and you also survive better to the spring for reasons independent of body size. The other explanation is that, of course, yes, the sex of the animal will determine its body size partly in the autumn, but it's body size, not sex, that's determining the spring survival. So how could you test this hypothesis using an observational control? Well, in the second graph, I've simply used a different color for the males and the females. So the females are in red, the males are in black. And if we look at this graph and we concentrate just within the group of males or just within the group of females, we notice that the correlation still persists. So when sex is fixed, when it's only males, when all of the individuals are male, so they're not changing, there is still a correlation between body size and survival. And similarly for the females. So this observation suggests then that if we statistically control for body size, and the second hypothesis is true, then we will still see a correlation. But if the first hypothesis is true, then once you've looked only at males or only at females, then there should no longer be a correlation between body size in the autumn and survival to the spring breeding season. So this is an intuitive example of replacing a physical control with an observational control. And again, the two are not the same, but I'm just giving you an intuitive example of how we might do this. Now let's imagine a little bit more complicated explanation. Imagine we think now that it's the quantity of summer forage, which varies from year to year, which determines the body weight of the individual in the autumn. So more summer forage results in a larger body weight in the autumn, and a larger body weight in the autumn then increases the probability of survival to the spring. So if you were to use the same logic as in the previous slide, then in order to test this hypothesis, we would have to be able to statistically control for body weight, hold it constant so that it doesn't change, so all of the organisms within that group have the same body weight. And if our hypothesis is true, then the correlation between summer forage and probability of surviving should disappear. The problem, of course, is that body weight is a continuous variable. So if we were to make groups of animals having the same or the fixed weight, then every animal would be in a different group and we couldn't look for any correlation. So how do we control for a continuous variable? Well, these topics, the relationship between physical controls and observational controls, and how you implement these controls are the topic of the next lecture. Before you go on to the next lecture, though, I suggest that you take a break now, go take a walk or do something else. Think about the topics that we've discussed so far. Try and apply them to your own research questions. And then, when you've been able to clear your head a little bit, we'll come back and we're going to have a rather longer lecture in which we're going to look at how we go about translating between the language of causality and the language of probability distributions, and how we can replace physical controls by observational controls, how the two are different and how they are the same. So take a break, and when you're ready, come back and listen to the next lecture.